Hello, everyone. Welcome back after the coffee break. Our next speaker is Christina Hvilsom from Copenhagen Zoo. Christina has been very active the recent years in researching genetic diversity, promoting genetic indicators into policy and management, and the perfection in the imperfection. I look forward to what is behind that. Thank you, Christina.
Thank you. So it's always difficult to come right after the headliner of today. But fortunately, you're filled with energy and some of you caffeine as well. And I'm really excited to get to be right after Robin, because it's a nice bridge to what I'm going to talk about, which is how we go from a theoretical concept as NE and make it applied. So let's dive in. So I'm assuming, well aware of the importance of genetic diversity, but also that it's one of the three pillars that kind of makes up biodiversity. And of course, it's important both to maintain and restore evolutionary potential, fitness, and ecosystem resilience. So without monitoring and also indicators, we cannot really follow the processes that threaten genetic diversity, such as loss of genetically distinct populations, reduced population size, human-induced selection, for instance, climate change effects, and the spread of alien populations. But what is an indicator? Well, it's a measure or metric, you could say, that indicates the state of something. It's the indicators for biodiversity is used to measure and monitor the state of biodiversity, identify both pressures and also threats, and assess effects of proactive measures and the resilience of and also restoration efforts. So I'm going to take you back in time a bit, because where we are now is an outcome of a lot of efforts that have been ongoing over the years. So this is genetics in conservation policy and management timeline from 75 to 15. And a lot of things have happened, both in terms of research milestones, but also in terms of policy. And I would predominantly be talking a lot about policy, but also going into the importance of research. So one of the things I would like for you to have a look at is CBD. So what is CBD? CBD is the United Nations Convention on Biological Diversity. And the first negotiations leading up to the CBD in 1993 were several different so-called articles. So there was an article about the identification and monitoring of biodiversity, but also one on in situ conservation, and one on the sustainable use of components of biological diversity. So this all has quite important effects for what came on later. Let's see, we do that. Good. So in those article texts, there were a lot of words being put into how to actually monitor and track the effects of conservation efforts and how to monitor them. So looking again a little bit ahead, because these were the first draft of the CBD. And over the years, there have been several meetings and events and new kind of frameworks, which I will be talking more about in a second. So another important thing that also happened was the IPBES, which is an independent intergovernmental platform for biodiversity and ecosystem resilience. And also there were under IUCN, the establishment of the Conservation Genetic Specialist Group, and also various other initiatives focusing on genetic diversity and resilience of ecosystems, such as Congress. So looking a bit further from 15 and up till around 2020, there were, again, different research milestones and policy level. There were a lot of things happening. There was both the Conservation Genetics Working Group of the Society of Conservation Biology and the Genetics Composition Working Group of GeoBon. And more recently, initiatives such as the Coast Action Network GBI, and also a kind of a network of networks trying to align some of the major initiatives looking at genetic diversity called the Coalition for Conservation Genetics. One important research effort I want to highlight is Linda Leiker and colleagues' paper from 2020 that stated that the post-2020 goals for the Convention of Biological Diversity overlooks genetic diversity. So I will come back to what that actually means. So if we look at the CBD, the way the CBD works is that they have meetings, biannual meetings. So every second year they have a so-called conference of the parties. And I can say that right now there's 196 parties to the convention. So it's predominantly countries and then the EU. So it's really globally that countries have signed up to this agreement. So back at COP2, so the second COP in '95, there was some wording in terms of that it was essential to identify the driving forces determining the status and trends of components of biological diversity. And fast forward to the COP3, the issues of indicators, assessment, and monitoring should be considered. In terms of the group working on these matters is the SABSTA, the subsidiary body on scientific, technical, and technological advice. It's nice. A nice long name. Again, fast forward a bit to COP6, there was further improvements. So a strategic plan for the decade around 2000 to 2010 to guide further implementation of the CBD. And there was also a decision on designing national level monitoring programs and indicators. And there was a need for establishing that. Fast forward again to COP8. A provisional list of global headline indicators for genetic diversity was on the horizon there. But when talking about genetic diversity, only exceed crop collections and the number of livestock breeds were mentioned. Then came the Aichi targets and the strategic plan for biodiversity in the decade from 2010 to 2020. And in that, there was the Aichi target 13. So that target addressed genetic diversity, but again, focusing on cultivated plants, farmed and domesticated animals, and their wild relatives, but no mentioning of wild species. So around 2020 and also a little bit before that, a lot of great minds put a lot of great efforts into highlighting the need to include genetic diversity of wild species in the CBD in their so-called global biodiversity framework. So there was, again, a lot of different papers pointing towards the neglect of genetic diversity of wild species. And these are just some of them. There was also some of these great minds also went to the COP15 in Kunming, the Kunming Montreal, held in Montreal, but doing various side events, having information booths, really engaging with the different policy stakeholders, doing a lot of lobbying, both prior to the COP, but also at the COP. Also, before the COP, 3 genetic indicators were proposed by this larger group of people, and it was 3 pragmatic indicators. These were not the only ones that were proposed, but this group of people proposed these 3. So it was indicator 1, which is the NE500, which you have heard about previously, but also indicator 2, which is the proportion of populations maintained within species, and a third indicator on the number of species and populations in which genetic diversity is being monitored using DNA-based methods. I would like to highlight that both indicator 1 and 2 can be assessed without genetic data. So it's both DNA-based but also proxy-based data. And then, fortunately, in 2022 at the COP15, indicators for genetic diversity for wild species was adopted. So it was the NE500 and the populations maintained that were adopted. So the NE500 is a so-called headline indicator, and the populations maintained is a complementary indicator. So meaning that all the signatory parties of CBD need to report on the headline indicator. So in the text of the Kunming-Montreal Global Biodiversity Framework, it says there is a goal and there's a target that has genetic diversity of wild species included. So the goal A towards 2050 focuses on ecosystem restoration to halt species extinction, abundance of native wild species increased, and does explicitly mention that genetic diversity within populations of wild and domesticated species is maintained, safeguarding their adaptive potential. But this is quite strong wording. And also, again, in target 4, this is a target for 2030. A target that focuses on halting extinction and recovery and restoration of species. And the wording says, as well as to maintain and restore the genetic diversity within and between populations of native, wild, and domesticated species to maintain their adaptive potential, including through in situ and ex situ conservation. So this was a huge achievement to actually be there. So this was 30 years after the CBD came into force. So it has taken a lot of effort and time and lobbying and talking to stakeholders, hearing what they have to say to get to the stage where we actually have indicators in the Convention of Biological Diversity. So I just wanted to mention this as well, because I know that Victoria is fortunately just after me, and she will give some very nice examples of how the practicalities of using indicators. So I won't dive into that too much, but I do want to mention that in order to get to the indicators, you can, as I mentioned before, use DNA-based, but also non-DNA-based data to get to those. But it all really starts with the EBVs, which is essential biodiversity variables, which are parameters, which is biodiversity observations at one location over time or several locations over time. And the NE, effective population size, is actually one of the EBVs, and the indicator is then the NE 500. So again, I think I will skip this because I think Victoria will repeat this quite a few times in terms of how to actually calculate the different indicators. So a lot of efforts went ahead of COP15, but a lot of efforts have also gone into the the post COP15. So I want to highlight these, and if you scan the QR code, it will take you directly to the relevant documentation. So the indicators are not perfect. We know they are imperfect, and there are a lot of pitfalls, and there are still scientific questions to be addressed. But they are usable for countries on the diversity of countries in terms of where they are in terms of resources, both within countries and also technologically, people, etc. So they are practical indicators that can be used, even though you don't have the infrastructure to do genomics on every single species in your country. But I wanted to highlight these because this is the description of some of the advantages of the genetic diversity indicators, but also limitations and how to overcome them. And then putting the indicators into use, so doing calculations for some of the countries. Again, not diving into this because I know this is one of the things that Victoria will tell you more about, but also pointing to this. So having a kind of a one-stop shop for all the material, the relevant documentations, guidance, etc., relating to the genetic diversity indicators can be found through this link. These are just some of them, and here are some of the additional ones focusing on the importance of genetic diversity, focusing on the indicators, how to put them into practice, how to adapt them into countries' strategic biodiversity strategies and action plans, etc. And speaking of that, so all of the countries that are signatory parties of the CBD need to report. And the first report, based on the Kunming-Montreal Global Biodiversity Framework, needs to be handed in next year in February, and the second one in 2019. So again, it's important that to kind of highlight where there can be improvements. So looking at some of the first biodiversity strategy and action plans that the countries need to do based on the CBD, Kunming-Montreal Global Biodiversity Framework. There is, again, there's room for improvement in terms of genetic diversity of wild species. So both writing scientific papers, but also producing policy briefs, because policymakers often don't have that much time. So kind of digesting that a bit, making it more easily readable and understandable for policymakers helps a lot if you want to convey a message. And another important paper is, again, down here. Scan the QR code and it will take you to it. I am going to tell you about GINEMO in a second, but I also want to say that it was not just going to the COP once and having the indicators adopted by the CBD, but actually also attending the COP last year to, again, follow up on how countries are doing in terms of actually adopting and using the genetic diversity indicators, and answering questions, offering guidance, etc. So, GINEMO. So there are several initiatives undergoing at the moment that focuses on both improving the science behind the indicators, but also helping to take that to countries and making them able to not only report to the CBD, but actually using genetic diversity indicators for monitoring, restoring, conserving their biodiversity. So GINEMO is Genetic Indicators for Nature Manager. Nature Monitoring. And GINEMO is a large transnational EU biodiversity plus research project. And it's we have a diverse team covering not only natural sciences, but also social sciences and others. And it's based on the need for countries to report to the CBD on genetic indicators. And GINEMO's mission is to co-create user-friendly workflows with country stakeholders that enables them to actually report on the Kunming-Montreal Global Biodiversity Framework, and genetic diversity indicators, and use them for biodiversity monitoring and action. And we are quite a large team. We are close to 30 people. We have partners from 11 institutions in 8 countries. And some of them are represented here today. And what we will do is that we will both determine best practices to obtain accurate and robust effective population size estimates for species with DNA-based data, but also develop best practices to estimate NE from proxies, so non-DNA-based data, and proportions of populations maintained with publicly available data sources. So we would like to address kind of the major factors that affect contemporary effective population size, such as kind of different spatial distributions, life history traits, genetic marker numbers, types, sampling schemes, estimation methods, etc. And using, simulations and proxy data and real data and the full shebang. And ending up with developing standardized workflows to facilitate the calculation of genetic indicators, both of them, in accordance with, of course, FAIR principles and open science practices. And again, also operationalizing the EBV workflow, so actually feeding essential biodiversity variables into the kind of EBV queue. So genetic indicators, as mentioned before, can be estimated based on both DNA-based and non-DNA-based data. So DNA-based, I don't think I need to say that it requires samples, it requires a lab, and it requires some analysis. And proxy data can be sourced from various sources, and I'll come to that in a second. So there is, for instance, census size, the NC that we also heard about just before, can be used as a proxy for NE. But again, looking into so the NE-NC ratio, for instance, is something that we're also going to look into. Examples for proxy data could be species population data, satellite data, which is one of the work packages, citizen science data, etc. And it should be easy to use with sufficient information. So potential sources for proxy data are species management reports and recovery plans, site inventories, flora and natural history books, global and local IUCN Red List databases, national species databases. You have that data banking here in Norway, for instance, scientific papers, collaborations with geneticists, consulting local knowledge holders, local NGOs, using citizen science data, habitat quality data, field guides, etc. So there is a plethora of sources, but of course there is to some of these sources also a trade-off in terms of the robustness of the estimates. So the behind there's both the scientific component, so actually calculating and testing the indicators scientifically, but also taking that and actually enable countries to report and use the genetic diversity indicators. So we have chosen an innovative methodology. So we are following a very inclusive stakeholder process with countries, a so-called co-creation process with training facilitators, where we work with the countries to understand what do they have in place at the moment, what are their resources, what are their challenges, and then taking that into account, working within the GENOMO project on those challenges together with the stakeholders to actually address these challenges and find solutions together with the country stakeholders so that we have an outcome, a product that they can actually use that fits their needs and resources, with the philosophy that if you actually address something that is a true challenge for the stakeholders, it's much more likely to be adopted and used. So these, we've run country workshops in 5 countries. So these are pictures from Belgium, France, Italy, and Sweden. And this is a picture from yesterday at the 5th floor of this building, where we held the workshop for Norway. So there are a lot of different challenges that came out of these workshops, but I do want to highlight just a few of them. One of the challenges that is common across all the countries is how to choose the species. There is a guidance to be found. There are IUCN guidelines on how to select species and population for monitoring genetic diversity. And there are case examples from countries and regions that have gone through a species selection process. But it is something that countries do struggle with on how to do that. There are some criteria, some basic criteria for selecting species, and then there are some criteria which can be adjusted based on the country's need. So this is something, but it's a process that the countries need to do themselves and reflect on which species they would like to use or would like to calculate genetic diversity indicators for. Another challenge that most countries that came up in the country workshops was how to define populations. And that too can be a challenge. The IUCN Conservation Genetics Specialist Group has had a project looking into how to define populations for genetic diversity protection. Unfortunately, we have the lead here today, Julia, is sitting up here. So if you want to know more about this project, you can either go and look at the preprints down here. There's also a paper on the genetic load behind the ESUs, or come talk to Julia in the break. So this is all good. The countries need to report a first time in 2026. So GNMO is working with the countries, some of the countries now, and we also have a focus on the reports that will be handed in February next year to see how well countries deal with actually getting calculations on genetic diversity indicators. But we don't do that alone. We do that together also with GENOA. So GENOA is a brand new EU cost action network. So it just launched in October last year, and the main aim is to implement genetic diversity indicators to broaden the uptake and use of genetic diversity for species assessment to protect biodiversity. It's a 4-year research network. So if you are interested, we are keen to have people from Norway in this cost action. So please have a look, access it through the QR code. It's also here in the button. And these are the 5 working groups of GENOA, which one of them on policy, which I'm leading together with John Hopen and Christina Hvilsom. And then we have working group 2, which is more about the science of the indicators. So it's Linda Leifer, Joachim Mercier. And working group 3, genes to ecosystems, Miriam Hertz and Ivan. And working group 4, engagement with Geno Sickelbacher and Elena Busan, and working group 5 on communication and collaboration. And if you want to know more about that, Alex is your guy. So please have a look if you would be interested in joining this network. But another I think could potentially be a game changer as well, is that some of us drafted a motion or text for a motion, which actually just got accepted. So now it's up for voting in terms of IUCN, and if enough support the motion, it will become a resolution for IUCN to work on. And the motions can be found online. And it's basically that we pledge for a task force under IUCN to be established to explore a standardized genetic diversity assessment, which is something that is missing in IUCN. So I just wanted to end, I think, my time is up, to say that this has been a huge team effort. There are so many initiatives over the years who have contributed and will continue to contribute. And a lot of different partners also that have helped push for the genetic diversity to be included in the CVD, but also continue to work on actually putting them to use on a country basis. So in summary, yes, we have indicators. We also know that they are not perfect. There is still work to be done. But they can be used no matter where you are, in which country you are in the world. They can be used both in terms of DNA-based and non-DNA-based data. Another thing I want to highlight is that, yes, I know it can be sometimes when you're a scientist and think about policy, you go, oh, but you can actually engage with an impact policy. And I think this is a prime example of that actually being done. But be mindful, don't come prepared, don't come alone, come with a lot of people. It takes a lot of effort to do it, but it can be successful. And then there are initiatives working on with countries on reporting and using genetic diversity indicators. And there are also initiatives that focus on the scientific work to improve the indicators, and also developing tools to ease the application of the indicators. So the last thing I want to stress is that countries are diverse. So these are 5 European countries, and they have various challenges, but there are also overlap in terms of challenges. If we look at kind of the complete kind of global view, we will have even more diversity and even a larger span in terms of resources in those countries to actually implement and use genetic diversity indicators. So there's still a lot of work to be done in order to put genetic diversity indicators into use. And I think that was my end slide. And I want to thank you for listening. And thank you to all the fabulous colleagues and collaborators on this.
Thank you, Christina. Are there any questions, quick questions in the auditorium? This one, Thomas.
Thank you. It's very interesting to hear this thing. As a scientist, not working sort of with policy, it's good to get sort of this input on what is actually happening. I'm wondering about this NE and these indicators and all of these different ways of estimating those. Will there also be some sort of indicator for sort of the quality of the estimates of the NEs?
That is the quality of the estimates. That is something that we are testing, of course, the robustness of it. But into the CPD, no. But of course, in the guidance, etc., that would be part of it as well. Yeah.
No more questions. I think regarding the time, we maybe move on and take a few more questions in the end.
Yes.
After Victoria.